My name is Nicolas Daviaud. I'm originally from France. I received my PhD in neuroscience since 2013, and then I came over to the US as a postdoctoral researcher at the Icahn School of Medicine at Mount Sinai, where I studied neural cell response to injury during neurodevelopment. Then I joined the TISH-MS Research Center in January 2019. When I came to the lab, Dr. Sadiq explained that one of the difficulties in MS research is getting patient brain and spinal cord tissue. So my first goal was to try to overcome this hurdle, so I decided to bring the mini-brain technology into our laboratory. We first collect cells from patient blood samples, and using the reprogramming technology, we can trigger that transformation into pluripotent stem cells. Those cells hold the capacity to differentiate into any type of cell of the human body. We can then, following a long protocol, transform those cells into mini-human brain. Because the brains aren't fully developed, we call them organoids. Those organoids retain all the genetic information of the patient they have been created for. So this technology has a great advantage. We can study the brain tissues during their development and determine if their genetic background can be an inner trigger of MS, or if it can lead to any difference in the disease progression observed in patients. We can also control the environment in which those mini-brains grow, and study how brain tissues react to different environmental triggers such as virus infection. This may help us explain why some patients have a difference in their disease progression and severity. Organoid research led to several breakthroughs in other diseases, such as send-off disease, autism spectrum disorders, or schizophrenia, among others. It has also led to a better understanding of the effect of Zika virus infection. Interestingly, it has never been done in MS. We eventually started this work in taking blood samples from LC controls and from patients with PPMS, SPMS, and RRMS. We have already developed these organoids to a certain extent, and now we are going to analyze the behavior of the different brain cells and to look at disease severity as well as factors that interact with it, like EBV infection. Although this research is early, we are already seeing differences between MS and control organoid, especially in the regulatory aspect of cell kinetics and development. Particularly, in progressive form of MS, the early data seems to indicate that certain cells are in hyperdrive mode. I'm really excited about this research because it is an innovative way of studying MS and may allow us to determine why, for example, in PPMS, brain repair mechanisms appear to be absent compared to RRMS. It also can help in trying to figure out the genetic contributions to disease development. For example, looking at identical twins, why one develops MS while the other one doesn't. Organoids may also be the best way to determine the role of environmental triggers.